Hi, my name's Matt Covey. I'm the University Librarian here at Rockefeller University. And today we're very fortunate and happy to welcome Sir Paul Nurse to talk about his book. We've been trying for a number of years to get Paul to come and talk about this book. Obviously, COVID has been a problematic feature in all of our lives, so it's been a long gestation, but we're really thankful he's finally here to talk about it. As most of you know, Paul is a past president here at Rockefeller University. He succeeded Arnold Levine in 2003 until 2011, when he took up the post at the Crick Institute, where he's the director, and also he's the Chancellor at the University of Bristol in the UK. So today, Paul is going to talk about his book, What is Life? The book was published in 2021, and it discusses 5 great ideas of biology, ranging from the cell to the logic of life. And understanding these 5 important concepts brings us closer to understanding the definition of life. So as I said, we are very pleased to welcome Paul here today to the Reader Marcus Fritz Library Annual Book Talk, and we look forward to hearing more about this book. There will be a Q&A after Paul speaks, and then a reception to follow upstairs in the main reading room of the library. So we hope you'll all join us once Paul's finished. So I would urge you once again just to welcome Paul back to a home away from home, and hopefully a great talk. So thank you, Paul, and I'll give it over to you.
Thank you. Well, hello everybody. It's a real pleasure to be back here, I have to say. It didn't take several years, I'm sure. I think it only took 1 year for me to get back. I really am pleased to be giving this talk, I have to say. I should emphasize it is a popular talk about a popular book. I don't mean it's necessarily popular, I just mean it's not scientific, if you understand what I mean. What I wanted to do was to write a book which you could read in 30 years and it would still be basically right. So in some ways, it isn't looking over the horizons, because most popular books 3 years out are basically not right, because they try and look over the horizon. So most of the things I will talk about will be very familiar to most of you in the audience. But I may have some interesting things to say about them that, that will put it together in a different sort of way. Okay, that's the introduction. I'm going to talk about the book, which asks one of the most fundamental questions in biology. You could argue the most fundamental question, and that is, what is life? And let me say at the very beginning, it's an easy question to ask, not such an easy question, in fact, to answer, believe it or not. And before you get too excited, I'm not going to give you a very good answer by the time we get to the end, but it will be one that I hope will stimulate your thinking about it. Now, there's an extraordinary diversity of life on the planet, as you all know. All around us are living things, bacteria, fungi, plants, animals, and of course, our cells. So what is it that links all these living life forms together? It's a long time ago, and it may be a bit metaphorical, but I think it was a butterfly like this one you're seeing here on the slide that first started me thinking seriously about questions like this. It was early spring. I was around 12 years of age, and I was sitting in our garden when this quivering yellow butterfly, well, not this particular one, but one of this type, this species, flew over the fence, turned, it flew a bit, hovered around, then a shadow disturbed it, and it took flight again, disappearing over the opposite fence. Very, nothing that exciting. But that intricate, perfectly formed brimstone yellow, that's what it was, made me think, because it was both very different to me, and yet somehow familiar. Like me, it was obviously alive. It could move, it could sense, it could respond. It seemed full of purpose. So I found myself wondering, what does it really mean to be alive? I was a bit of an odd 12-year-old, by the way, I ought to say. And that is, of course, the question, what is life? I'm obviously not the first to have asked that question or the first to write a book about it. For example, this is the title page of probably the most famous book called What is Life, written in 1944 by the famous physicist Erwin Schrödinger, who wrote only one work on life. He was a physicist, of course. His book was influential, including for me. I first read it over 50 years ago. And I recommend you read it now. He did look over the horizon, and he sort of got it right. Not completely right, but he sort of got it more or less right. The approach, as you've heard, that I have used in this book is to consider some of the most important ideas, at least for me, of biology. I describe where they come from, and one thing I want to emphasize, it's surprising how the key ideas of biology are often rather old. We tend to think everything is sort of invented in the last 20, 30 years. That isn't the case. And I want to explain a little bit where they came from. And then at the end, I will pull it together and try and develop a set of principles that are fundamental for thinking about life. Now, the first of these ideas is the concept of the cell. The cell is the basic structural and functional unit of life. I'll describe what I mean by that in a moment. I like to think of it as life's atom, you know, the basic unit of life. They were discovered by Robert Hooke in 1665 in the city where I live, in Oxford, in fact. We have a plaque on the wall saying he discovered cells there. And what he did is he took a razor and cut slivers away from a piece of cork, and then examined them under an early microscope, like the one that you see here. And what he saw were a neat array of boxes. He called them cells after the Latin word for cella, for small cubicle. Shown here in his drawing, the one in black and white, published in a very famous book of the time called Micrographia. It was a really big cellar. I put here a scanning electron microscope just to the right of it, taken about 15 years ago, and you can see it's really no different from what he drew in the early 1600s. So these were the first pictures of cells. A few years later, a Dutch draper, Antony von Leeuwenhoek, working in Delft, scraped between his teeth and put it under a microscope, single lens microscope, and discovered a single-celled life, bacteria. He sent this drawing to the Royal Society in London. I've actually held it. He sent about 1,000 such drawings, in fact, in the 1670s, 1680s. I quite like this one. It's rather charming. If you look at figure B to C to D, the bacteria was obviously swimming about underneath the microscope and did a sort of loop. He was rather, however, disturbed by the discovery of life because he was rather proud of his dental hygiene, and finding something, a microbe living on his teeth, really upset him, as he said in his letter. Now, he was a close neighbor in Delft to the painter Vermeer, and unusually for a 17th century artist, or for that matter, artists of almost any time, he painted a picture of a scientist. In fact, two pictures of a scientist, one in the Louvre, one in Frankfurt. I forget where this one is. And one's called an astronomer, one's called a geographer. It's the same person. And I like to think this is Leeuwenhoek. I have to admit to you, there isn't really any good evidence for that particular speculation, except when Vermeer died, Leeuwenhoek was the executor of his will, and they did live only 2 or 3 streets away from each other, and I think it's a nice story. So maybe we are looking here at Leeuwenhoek. Over the next 2 centuries, it became clear that all living organisms consist either of a single cell or, like us, a collection of cells. This conclusion was summarized by the German zoologist Theodor Schwann, in 1839, he wrote, "All organisms are composed of essentially like parts, namely of cells." A rather serious-looking gentleman, I think, German professor, of course, who said this. But as well as cells being the basic structural unit of life, which is what that's implied, they are also the basic functional unit of life. And this was also stated by another German scientist, in fact, the German pathologist Rudolf Virchow, the founder of pathology, essentially, and also of the origins of one of the origins of thinking about disease. In 1858, he wrote that a cell is the vital unit bearing the complete characteristics of life. That is, of course, that it's the basic functional unit of life, so structure and function. He also said all cells come from cells, omnis cellula acellular, for those who know the Latin. Although he wasn't the first to say that, it was said by others in the decades before him. And what this means is that all cells arise through the division of pre-existing cells, which wasn't actually universally accepted at the time because of spontaneous generation. People thought maybe life just sort of came out of dirty ponds or something of that sort. But I want to come back to this fact that all cells come from the division of pre-existing cells at the very end of my talk, because there's something more interesting about it. Now, I hope I've persuaded you that cells are interesting. If I have failed to convince you of that, then let me show you this picture here and remind you all, this is of course an egg, a mammalian egg being fertilized by some sperm. I'll remind you, all of you in this room were once a single cell like this, all of you. And if you still are not interested in cells, there are several doors you can leave now. I won't be offended, okay? But we all looked like that. Amazing, huh? We were all a single cell. Cells are considered to be the simplest entities which unambiguously exhibit the characteristic of life. I say unambiguously because I obviously have to talk about viruses eventually, and so I'll slither around that a little later. Now, this means that understanding cells is key to understanding life, and so they will feature quite prominently in what I have to say. Here is a cell, modern EM, artificially colored. They're bounded physical entities separated from the environment by those boundaries of a semi-permeable lipid membrane, which, although separated, they can communicate, of course, with their surroundings. This separation is important because it allows order to be built up inside cells at the expense of increasing disorder outside cells. And therefore, life doesn't contravene the second law of thermodynamics, which often upsets physicists thinking about this particular problem, because, you know, it's like the sacred law of the second law of thermodynamics. But there isn't a problem. Schrödinger summarized this up in this quite nice sentence, but he was obviously thinking about this. An organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. Beautiful sentence, but it means that a cell is not subject to the problem of the second law of thermodynamics. He also, in thinking about this, proposed that life had a code script. And this is seen in the, this was written in 1944. These chromosomes contain, in some kind of code script, the entire pattern of the individual's future development and functioning. And this introduced the notion of code script. It's implicit in what I'm about to say yet, but this was very clearly stated. So it's an introduction of the second idea I want to share with you, which is the gene, examining the mysterious phenomenon of inheritance. Now, this is the idea of the gene as the basis of heredity, of course. which had its origins with the work of the Augustinian monk Gregor Mendel, who was abbot of Brno Monastery, and he published 2 papers in 1866. Let me already note, we're back in the 19th and 17th centuries, with all of this stuff. This is Gregor Mendel when he was abbot. He tackled the problem of inheritance by carrying out crosses of pea plants with different characteristics and analyzing the outcomes in thousands of the progeny in these crosses. I found a picture of his garden in 1981 when I visited the monastery in the middle of the Cold War. It was actually extremely difficult to get to Brno, even more difficult to get to the monastery, and even more difficult to discover Mendel's cupboard where all his materials had been kept, and they were being kept away. Those of you who know about this, in the other side of the Iron Curtain, Lysenko was prominent, and Mendel was absolutely not. One thing that, you see this this garden here. This wasn't a small. This was a research garden, and Mendel also had a greenhouse of similar size, which has just been rebuilt. It was down when I was there in 1981, but I've seen it more recently, and it's actually quite a big activity. And the strange thing, and we don't think about this now, this monastery was basically a research institute. It was a sort of prototype Rockefeller, if you like, because the monks there they were a teaching order, and they each had a sort of scientific interest. Gregor was interested in plant breeding, which a number of other scientists were to improve crops, but he was also interested in astronomy. He made a map of the moon, meteorology, he plotted the changes in air pressure and so on. And there were others doing all sorts of other projects in this monastery. And it wasn't just a sort of little hobby. It was obviously very, very serious. We tend to think of research being done now like we do in universities, institutes, and so on, but it was in unusual places then in the 19th century. What Mendel did was crossed peas of various characteristics, plants of different heights, flower colors, and different seed shapes. And I want to say something a little bit more about this. He did a number of preliminary experiments with about 6 other plant species and could make no sense of them whatsoever. They were multigenic inheritance, so it just didn't make any sense. The only ones he could make any sense of were pea plants, because a number of the characteristics were monogenic, so it was much more straightforward. So being a good scientist, he abandoned everything that he didn't understand. It's very important to do that. Abandon the things he didn't understand and focused on the system which he could understand. And then when you understand that, you've got a chance of actually understanding the things you don't understand, which is something we need to teach our students a bit more. It's all right to admit you don't understand, and it's all right to cherry pick what you decide to work on, as long as you realize what you are doing, okay? And sometimes we can get a bit sort of pompous about these things, but you work on things that work, yes? If you don't want to go mad, anyway. Okay, so he famously observed these simple ratios in characteristics in the progenies, ratios like 3:1 and 9:3 to 3:1 and 1:2 to 1, and all of this stuff that we learned at school. And this eventually led to the idea of Mendelism, that heredity was based on pairs of indivisible particles or factors inherited from the germ cells, from the pollen and ovule in plants, sperm and eggs in animals. He didn't quite get to Mendelism, though it's named after him. If you look at I visited the monastery a number of times and handled, in this case, his original manuscript. And the notation in it indicates that it isn't quite as we would now understand it. And it took the discovery of chromosomes 30 years later and a rediscovery by 3 plant geneticists in the early 20th century to actually invent Mendelism, which they did name after Gregor. So these factors, because genetics is essentially, Mendelian genetics is essentially a particulate theory. It's a particulate theory. That's why you get simple ratios that come out. They were found subsequently to be linked to the chromosomes that were discovered after Mendel's death, to be separating in dividing cells. Here's a late 19th century drawing of onion root tip cells. And I have to tell you, the first time I saw cells was in onion. We're still doing this. I see you're nodding. I mean, we look at onion tip cells in roots and see chromosomes. You know, it's what I did at school in the 1960s. Anyway, what's important about this, I think, is that the observation that you have chromosomes separating and turned Mendel's sort of abstract thinking into a concrete reality. And that's when I think it became more convincing that there was something happening, because the abstraction of the numbers became real when you actually saw a physical interpretation of it. And that's something else for us to think about when we're doing science, of course. These Mendelian factors are, of course, what we now call genes. And in the 1940s, Oswald Avery here in Rockefeller University, right here, showed that genes were made of deoxyribonucleic acid. Not that anybody else in Rockefeller believed him, of course, at the time. And in fact, he had furious rows with people about that. But it was an amazing piece of very, very careful biochemistry in the 1940s. Here is a DNA, a sort of cartoon. You go from the cell, the nucleus, the chromosome, through to DNA structures and on to the double helix, the structure of which was established about 10 years after Avery in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, and in Cambridge by Francis Crick and Jim Watson. Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases making up the DNA and the pairing between complementary bases, which you see here with the sort of this ladder. The double helix is like a twisted ladder. It's easier to understand what's going on if you in your imagination untwist the ladder. So it is just a ladder, and then the ladder separates, and you've got complementing bases, A with T and G with C. Now, when these complementing bases, which are linked to each other, when these links are split, the two templates are formed, which then can be used to make two identical helices. And this explains how the DNA is copied during cell reproduction, to link us back to our first idea, accounting for heredity. And it also explains Schrödinger's code script and the permanence of the gene between generations, because it's constantly being copied, the code script, every time a cell divides. It is a beautiful idea. Mendel's stuff is beautiful. The structure of DNA is all. I get sick of seeing sculptures of DNA in research institutes. I tried to ban them in the Crick, and somehow it crept in, actually. And then I made a joke about it on the radio, and the donor of it got really cross with me, because I said I was trying to find a way to get rid of it. And sorry, Marnie, it didn't work, that particular... if you'd been looking after me, I wouldn't have had that. You'd have told me to be quiet with my jokes, but anyway. Crick went on to propose what is called the central dogma. Crick himself was quite an interesting character who I knew moderately well. Only he would have the sort of confidence to say the central dogma, you know. Do we talk about central dogmas when we discover things? I think not, anyway. And that explains how DNA encodes the structure of proteins, as you see here. It describes a sequence of bases making up the DNA. Is copied to RNA, ribonucleic acid, that acts as a messenger between DNA located in the nucleus, where the chromosomes are, and the cytoplasm, where the proteins are made. And the first approximation, there is one gene encoding each protein. Not exactly like that, but more or less like that. These discoveries laid the foundations for molecular biology, and I feel privileged, actually, of having had my entire life in the sort of production well, not me personally, but as the production of this understanding. And they are critical for understanding life. Central to it is the genetic code. It's written in a 4-letter code of the 4 bases, adenine, thymine, cytosine, guanine. And the information, and I'm going to return to this, and the information is stored in a linear form. This way of storing information is the same as the sentences I'm speaking to you. That's a linear form of, and also digital. The sentences that you read in a book, or, of course, bytes in a computer. It's all a linear storage of information. And it means that the linear digital storage of information was invented by life over 3 billion years before the computer age, just to put us in perspective. The nature and function of the gene introduces the next 2 ideas: life as chemistry and life as information. Let's start with life as chemistry. That is, we explain life in terms of chemical activity and physical forces. The origins of this idea can be traced back to Lavoisier, the father of chemistry, in the 18th century. But he was studying fermentation. He unfortunately lost his head in the French Revolution, which obviously curtailed his research career before he got tenure. But because he was a tax collector in the Ancien Regime and fell foul of Robespierre. But it was Louis Pasteur who carried out experiments on sugar beet fermentation. So this is clearly a French activity based on the study of fermentation, making wine, of course, so we can sort of understand it, which led to the concept that life is chemistry. This is a picture, I think it's in Paris, I forget where. Pasteur was analyzing the fermentation of sugar beet to make alcohol, and he was doing it at the request of industrialists because fermentations quite often failed, and instead of making alcohol, which is what they wanted, it would make acid instead. And the industrialists asked him to do a bit of applied research to see if he could correct this. And what Pasteur did, largely by looking down a microscope at what was in the fermentation liquid, he demonstrated or established that successful production of alcohol required the activity of yeast, a single-cell living organism, yeast. And he proposed that it was the yeast cell producing substances that made alcohol from the sugar, which was derived from the sugar beet. And if the yeast was not there, but bacteria, then the fermentation made acid. And that helped the industrialists, you know, because they seeded it with yeast. I don't know if he got paid for it. I've not actually found that out. I suspect not. Maybe got a free bottle of wine or something. But anyway, what Pasteur, being a brilliant scientist, thought about that, and he concluded more generally that fermentation was a physiological act yielding products for the cell, and that chemical reactions are an expression of the life of a cell. In other words, life is chemistry. There, 1870. But it has to be said, it's a very elaborate and special chemistry. It's based on carbon polymers, as are DNA and RNA, of course. And I now want to consider the carbon polymers that make up proteins. Because the substances from yeast that Pasteur postulated, which made the alcohol, are proteins that act as enzymes which catalyze chemical reactions. Now, proteins are polymers of amino acids with a backbone of individual amino acids. You've seen that here, and side chains of amino acids. Now, a structure of this is shown here, and what I just want to illustrate is just a sample of the 20 natural amino acids that we have. The chain is very similar. That's the bottom. It's a sort of carbon, carbon, nitrogen, carbon, carbon, nitrogen. But the side chains, which are the individual amino acids, are chemically different. Some are positively charged, some negatively charged, some repel water. Some are big, some are small. And these are the 4 or 5 of the 20 that are shown. This is another picture of it, a structural one. Has some structural biologists in the audience. They always like looking at angstroms, so I thought I should put that up there, which is the same as what we've seen. And of course, amino acids make chains. The chains fold up depending on the chemistry to make structures. And here is a billiard ball type structure. It's been covered, which represents the different properties of the amino acids. So now what you have is 3-dimensional structures with quite different parts of chemistry embedded within it. And it's this which makes it so more interesting chemically than DNA. DNA is a very good storage of information, but it's immensely dull. Whereas proteins, of course, are chemically extremely elaborate, and because of all the different folds and so on, that they can produce extraordinary chemistry from these folded up proteins. And it's this that brings about the wide diversity of chemical reactions that enzymes carry out. And enzymes and other proteins therefore act essentially as chemical machines in the cell. They produce a huge range of reactions and chemicals needed for living organisms to function. They make new molecules, they break molecules down, they recycle them, they make large molecular assemblies, they make nucleic acids, protein, lipids, and so on. They also do physical work. They make motors, they carry cargoes around the cell on protein-based trackways. They act in hybrid ways, combining chemistry and physics, capturing energy from sunlight to produce energy-rich chemicals that drive activities around the cell. And all this extraordinary chemical activity, and I'm going over the top here because we just don't, it is extraordinary range of chemical activities, is all going on simultaneously in the tiny space 5 microns by 5 microns by 5 microns of a cell. And a small sample of these reactions are shown of what some of us had to learn at university. This is a map of the central metabolism of the cell. For those of you who had to, I hope you don't, nobody here still has to learn this, but the red circle is the Krebs cycle, if you're at the bottom there, for example. Now, I want to make a few things about this, which we don't normally sort of think about. These reactions essentially require somewhat different chemical conditions for them to actually work. They had to be separated one from another for the system to work. It's achieved by compartmentation brought about by the surfaces and folds of the enzymes, complexes of enzymes together, acting together, membrane-bounded organelles, as we see in the cell, and schematized here in something I probably stole from Scientific American or something, where you can see the different chemical, where you can see the different compartments, which I'm trying to emphasize are chemically different compartments. There's different things going on to make it all work. And this allows the hugely elaborate chemistry of life to actually take place. But for this to all work together for a functioning cell, this chemistry has got to work together as a whole. And this is what the real mystery for me of life is. It has to be organized together, which requires communication between all these different chemical micro environments and compartments. Communication is the transfer of information, and that's the next idea I want to talk about, which is life as information. We dealt with life as chemistry and physics. Now I want to consider complex systems, control, purpose, because these two, this is what we really need to be thinking about, the interaction of these two systems. Now, life is complex, do you agree? And I wanted to find out who first discussed that, and it took me a while to track it down, and I got some help from somebody then at Sussex University. But you'd be surprised to hear that the first person who talked a lot about this was Immanuel Kant, the philosopher Immanuel Kant. And he published a number of books, and I think it was one on moral philosophy. It has about 20 pages talking about life as a complex system. And of course, That was in 1800. I hadn't the slightest idea what it had to do with moral philosophy, but it is really quite interesting, and that is where its origins are. Now, what do we mean by terms like the management of information? Well, I'm going to give two simple examples. One everybody's familiar with. The second, most of you will be familiar with. Now, this is back to DNA, double helix. The structure of DNA is iconic. It is beautiful. But its real beauty is not the structure. It lies in the fact that it only makes sense when it is recognized, as I've already argued, as a digital information storage device. That is what is important for life. One written in this linear script of 4 letters. It's ideal for storing information with the encoding letter bases turned inside the helix, somewhat protected. And this is the important point. This information through the proteins can be turned into chemical action that can do work through all those varied chemical activities. So you have DNA that stores the information, and then that information is transcribed and translated into chemical activity with the proteins. And we don't often sort of think of it, at least I wasn't taught of it like that, but that is really the, one of the most beautiful things about life. So the ability to encode information in inert or fairly inert one-dimensional linear structures, although I've just been to my lab and the DNA has all been degraded, so it's not all quite as stable as we sort of want. And the ability to carry out chemical and physical work in active 3-dimensional structures is a key feature of life, and one I think needs to be emphasized. Second example, with respect to information, is regulation. That is making the complex living systems in living organisms to act as a whole, to act with purpose. A familiar example of regulation is found in man-made machines. Now, I don't know if you're like me, I understand most machines made up to about 1900, and increasingly not as the years go on past that. And so what you see here is a governor developed by James Watt to control steam engines that I took a picture of on a steam engine made before 1900. And it's simple. We can understand it. As the engine goes faster, the balls swing out, right? It shuts off the steam. When the spindle goes slower, they swing in and they switch on the steam. This leads to homeostasis, remaining a constant speed of the steam engine. So it's a controlling system. The governor is an example of a negative feedback loop. And if we look at it in the context of metabolic pathway in a cell, it's a negative feedback loop. You see at the top there, A makes B makes C. As C builds up, it inhibits A to B, and as a consequence, C can then fall down if it's going somewhere else. You can have other sorts of feedback controls. Below that is a positive feedback loop where A to B to C, C speeds up to A to B. So this acts as a switch. It acts as an irreversible switch. These ideas were first demonstrated by the brilliant French geneticists Francois Jacob and Jacques Monod, who didn't do any chemistry. They just counted colonies of bacteria on plates and thought about it. And I often say we don't think quite enough. Now, we need to think about what these things mean. And they worked out the famous lac operon and the regulation just doing genetics on bacteria. Now, control modules lead to these different types of negative feedback, positive feedback, and there's many different feedbacks and control modules operating in cells, bringing about homeostasis, generating switches, timers, toggles, oscillators, all translating chemistry into informational modules. So that's a sort of idea. Chemistry is being turned into informational modules. They can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for this is an electronic circuit, which is all linked modules. Again, I've stolen this, I think this time from Embo Journal, if I remember, about 25 years ago, and some wit took a transistor radio and then put oncogenes on it to show that it was all part of a complex network. in reality, an even better metaphor is not hardware, but what has been called wetware. Because in living organisms, the components can be relinked in different ways through solution chemistry, so that you have the ability to change the networks, to change the hardware, because it's not hard work. So this allows, ultimately, for the complex system to act as a whole with overall purpose, reproducing a cell, making a butterfly, maintaining our cells. Now, for generations, it was thought that purposeful behaviors required a designer, a creator. And it took evolution by natural selection to change this, and this is my final idea. And if any of you are beginning to flag, this is the last idea. I will then only talk about viruses, then I'll bring it together. So if you're having trouble, deep breaths, it will soon be over, okay? This is probably the most beautiful idea, really, in all of biology, evolution by natural selection, mainly due to Charles Darwin. It has two parts, that life evolves over time, and secondly, the major mechanism, or a major mechanism, to quote him more specifically, for evolution is natural selection. He's much more open than people often think. He didn't know what else was there, but he imagined there could be there. Now, the first of these ideas that life evolves over time was not actually Charles's idea at all. It had been talked about for up to a century, particularly by individuals like Jean-Baptiste Lamarck, famous for Lamarckian ideas, who's been underestimated because it was very sensible what he wrote. And Charles, his own grandfather, Erasmus Darwin. The one on the right was the serious Victorian Charles Darwin. On the left was equally looking serious, but I'm going to say a few more words about him, is Erasmus Darwin. Now, Erasmus Darwin was a much more colourful character than Charles. He was a scientist, he was a doctor, and he was a poet. And he wrote all his research studies up in the form of poetry. I have several of his books from first edition from the 18th century. And it's really true. You know, he'd write a book called The Love of Plants and talk about plant breeding and so on. It was all in blank verse. Okay, he was sort of rather overwhelmed and forgotten by, you know, when Wordsworth and Shelley and so came on the scene. But he was actually quite well respected. He was also a Republican. He refused to be George III's doctor about 3 times because he was the most famous doctor in England. He was a proponent of women's public education and wrote a book on what girls should be taught at school. He set up a school for girls. The teachers there were his 2 illegitimate daughters, by the way, but he was colourful in other ways as well. And he was a great conversationalist, despite having an appalling stammer. And the other thing that I quite like about it, he was absolutely nowhere close to a university. He was doing all his work in the Midlands. He was miles and miles away from Oxford and Cambridge. And he was part of the Lunar Society, which involved a whole variety of engineers and some scientists that met once a month under the full moon, and so got a term as the lunatics. So they called themselves the Lunar Society. It's very colourful, all this stuff. It's sort of, sometimes I think we're rather boring today, do you know? Anyway, pass over for that. Now, Erasmus and Lamarck were saying, life has evolved, but they didn't have much evidence for it. Whereas what Charles did was to collect data for it. And of course, that's the other side of science. Now, the evolution of life implies there's a tree of life. That is, living organisms are related. And if life only arose and survived once on the planet, which is quite possible given the conserved chemistry and coding, then all life on Earth is related to each other. And that's an important point I want to leave in your mind. This connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859, where he illustrated a tree of life. Look at the bottom there, though, A to L. He obviously wasn't sure whether life arose once or many times, and he sort of leaves that sort of blank. And of course, we don't quite know that either, but it may be that life as we know it only survived once by outcompeting others. He found evidence in favor of life evolving through his time on the Beagle for 5 years, and he proposed natural selection, which was primarily his idea. There were actually one or two others who had some related ideas to explain how evolution might take place, based on natural selection, which is a consequence of populations of species exhibiting variations caused by inheritable genetic changes. And these can be caused by changes, mutations in DNA and damage and so on. And the important thing about this is that it produces variants but behave differently. And if these variants influence characteristics that make organisms more successful, better adapted, they reproduce more. And as a consequence, that particular genetic variation will spread through the population. Such a simple idea, but actually does explain how you can get adaptation and therefore apparently design. When you collect together enough genetic variations, then it's possible the new population will no longer be able to breed with the original one, and that's why it's the origin of species. That's because it explains species. Darwin came to this view in part due to his studies of finches on the Galapagos Islands. That's on the left here. And what he did, he described these finches, which had beaks evolved for particular different purposes. You know, the one at the top breaks nuts, the one at the bottom feeds under bark, trying to find insects. Just like the pliers on the right are designed to do different things, but obviously related. But the difference is, of course, is that on the left they evolved by natural selection, and on the right they were intelligently designed. Now, this is still contentious for some, and is, of course, particularly those who feel that it has to have come about by a divine designer or creator. Now, for natural selection to take place, 3 things are needed: reproduction, hereditary system which exhibits variability. And I'm going to give you a very simple example of this, just so that you get it, all right? I want you to imagine a single-celled life form with a brown cell coat, which is determined by a particular gene. Imagine there's damage to its DNA, so now the cell makes a red coat. Also imagine that red-coated cells survive better. Perhaps they avoid predators or something. As a consequence, red cells replace the brown cells, and bingo, evolution by natural selection has taken place. Dead simple, of course, not like making an eye or an ear, but the same principle. This is a profoundly important idea in biology, so much so that the 20th century geneticist Hermann Müller used it to define what life is. He defined life as living things which have properties that allow them to undergo natural selection and therefore to evolve. And that is going to be central for what I'm going to say in a couple of minutes. Before I do that, I'm going to say goodbye to Darwin as an old man with his final monumental sentences from The Origin of Species. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Why can't we write things like that today? You submit a paper, anything interesting like this, you know, the reviewers say, oh, this is speculation. The editors say it's too many words, and it's gone. And you can't, you know, you read your papers a year later, and it's awful. You can read this now, and nearly 200 years later, and it's interesting. Note he was relating to the great physicist Isaac Newton, where he referred to gravity. That was not an accident, I'm sure. He wanted to explain biology has fundamental laws just like physics. It's not just describing all the different species of beetles or all the different sequences of genes, which is our modern version of it. It is, of course, establishing laws. Now, before I finally sum up, I need to have a little chat about viruses. This is a virus. It's a bacteriophage. They have a genome based on DNA or RNA. They undergo evolution by natural selection, so they would pass Muller's test, but can only reproduce when they are inside the cells of other living organisms. And they do that by hijacking those cells' molecular machinery to copy themselves and make more viruses. This means a virus is completely dependent upon another living entity. And this is why there's so much discussion about it. But in thinking about this, I think it's useful to remember that other forms of life are also, to some extent, dependent on other living beings as well, which is sometimes we sort of forget. The many parasites that live inside cells or bodies of animals are clearly an example. The extent of their dependencies are less than for a virus, but they are still dependent. We are dependent on living organisms. I mean, we eat, you know, what we eat. We have microbes in our body that make amino acids that we can't make. So, therefore, it means there's a graded spectrum of living organisms. They have different and varied dependencies on other life forms. In the case of the virus, that dependency is very strong, whilst in other organisms, it can get weaker. But I would say that's a gradation. It's not a principle. I argue that these different life forms are alive largely because of Muller. They all evolved by natural selection. But what that means is that life on Earth is fundamentally connected through two things: by us all being related as a result of evolution, and secondly, these deep relationships and interdependencies. Now, I'm now going to sum all this up, okay? But as I told you, I'm going to fail and not give you a dictionary-like definition, but I am going to say what you should be thinking about, okay? What is life? Living things are independent, bounded physical entities. They're separated from their environment, but in communication with it. This allows order and organization. The basic unit on life is the cell, and multicellular organisms are aggregates of cells. Next one. The central principle, I want to argue, is Muller's. Life forms are entities that undergo evolution by natural selection. By doing so, they acquire purpose. Some of you may want to ask what I mean by purpose, but they acquire purpose. They are entities that build, maintain, and reproduce themselves. They can evolve because they have the attributes of reproduction, they have a hereditary system, and that exhibits variability. Third set of principles. Life forms are chemical, physical, and informational machines. I emphasize the informational machines as well as the physical chemical machines. They construct their own metabolism. They allow themselves to build, maintain, grow, and reproduce. Life on Earth is based on carbon polymer chemistry. If we apply those principles, that wouldn't necessarily have to be the case. But carbon is very good for this because of its chemistry, and they can make these informational storage devices. They can do chemical and physical machines. Life is based on information management, encoded the information in a linear form, and living things are connected and related by common descent and close interconnections. Now, I think, should we ever encounter life elsewhere in the universe, I suspect it will be built on these principles. The chemical details could be different. And unfortunately, from a long distance, we can only look at the chemical differences. We can't look at some of these other things. It might not be based on carbon. But I do think to satisfy the need for both long-term information storage and also chemistry, it is probably based on polymers, because that is the ideal type of molecule that can do this. But I'm going to end by returning to our planet, because this is the only corner of the universe where we know for certain life does exist. The life we are part of is on Earth it's got bewildering diversity, as I started, and understanding it makes a fundamental contribution to our culture and civilization, as does the research at Rockefeller doing exactly that. It's creating culture and civilization. Our growing understanding of what life is has great potential to improve our lot. But the knowledge goes further. Biology shows us that all living organisms are related and closely interacting. We are bound by this deep connectedness to all other life, to bacteria, to yeast, to pea plants, to this yellow brimstone, all of which I've talked about. Now, together, all these species are life's last great survivors, the latest descendants of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. I wanted to emphasize that because you remember I started off with cell division. We're actually physically connected by repeated rounds of cell division, which goes back hundreds of millions of years. We're connected to the earliest life on the planet through repeated cell divisions. And when I sort of started thinking about that, I mean, it's astounding, this sort of connection. However, as far as we know, we are the only life forms who can see this deep connectivity and reflect on what it means. And I argue that gives us human beings a really special responsibility for life on this planet, made up as it is by our relatives, some close, some distant. We need to care about it. We need to care for it. And to do that, we need to understand it. And that's really what life, understanding what life is all about. And thank you very much for listening to me. Questions? I'm a bit deaf, so you have to shout, or you can interpret. Or we can go and have wine and cheese instead, if you like. Is there somebody brave? Hello.
Hi. I'm not sure exactly how to ask this, but it feels to me like the list of principles you offered are sort of a mixture of implementation details and principles. And I'm not sure. I'll give you an example. One example is cells. There's many examples in biology where there are syncytia instead of cells, like in Drosophila development or in the placenta or in certain algae that are giant unicellular algae. And then they become cells at some point in their life cycle, and and so on. So how are we to, like, if we're looking for principles, how are we to differentiate between the implementation details, like it's DNA and not some other polymer, and and sort of the core ideas, like where the cell, I think the core idea is boundedness.
Yeah.
Something like that.
I can't fully answer it, but I'd say we have been potentially misled by physics, which has different sorts of principles, which can be distilled in a way that we've got comfortable with because of the nature of what they are. Biology is a much messier thing. And I think it's actually best just to admit it's a messy thing. And in what I've said, I've mixed up, and what you're saying quite correctly, different sort of categories of things. I think that's what has disturbed you a bit, whereas that doesn't happen quite the same way in physics, although in some parts of it, it does. But I think we should just not get too uptight about it, to be honest. I think if it gives us some deeper meaning than just sort of describing what it looks like, and describing endless chemical reactions without thinking, what does it all mean together? I think something from it emerges, which is illuminating. And in writing this book, and I mentioned some of these things on the way, things cropped up which I'd never thought of before, which I think are interesting, and I'm going to elevate them to principles to make you think about it. But I wouldn't like to say that they are a special category of event, which is different from other categories. It's a bit of a cop-out, that, but I mean, you get what I'm saying. One here somewhere.
I was hoping that you would share your thoughts about how certain cells remain alive and maintain their function for very extended periods of time.
Mm. Well, by doing all the stuff I told you about. Basically, what we have here are things that underpin much, maybe all, but certainly much of all the different things living things do. And if you sort of think about it, you can couch it in... I think the central thing is life as chemistry and physics and life as information, which is brought about by natural selection to produce purpose. But the sort of thing that you were just saying, I think you can sort of distill it into that sort of framework. And I'd say more than that, I think we ought to spend more time thinking about that. We tend to get into the weeds a bit. You know, you pick up nature, you're in the weeds very quick, you know. And sometimes it helps, you know, to swim more freely. And not only that, you know, if the amount of information we're gathering now, description, sorry, of biological processes and so on, and we teach our students, we're going to piss them all off. I mean, they're going to just have so much information, you know, that we've got to distill it into something that is interesting, or we're going to lose them. Yes. Ah, now we're getting there. Right. Wine and cheese is receding.
Thanks. Yeah, I was going to ask if a computer operates along these principles, would you call it alive?
No, I forgot my cop-out sentence there. I think which was that it has to have a physical basis, which is itself self-organizing. So you can make computer games, of course, which evolve by natural selection, but they don't have these other properties. But we learn something from the computer games, and we learn something from the neural networks and so on that we see increasingly being used in, you know, the machine learning of artificial intelligence. So I think it's informative, but I don't think it's worth going down the cul-de-sac. Is a computer alive? You know, you can just switch it off by turning off the electricity. You know?
You mentioned Schrodinger's book, What is Life? And there's another one between yours and his, which was Lynn Margulis's book, What is Life?
Yep.
Of course, she got part of the story of life right, the endosymbiont hypothesis, but also took a really philosophical turn, and her book is very philosophical. So did you read that and think about whether there was anything worth talking about with respect to more philosophical aspects?
I have read it, and her, you know, suggestion about the symbiosis is of course very important, and she had a hard time selling it initially. It is, of course, only one aspect of life and of essentially eukaryotic life. So it's very important. I tried to keep away from philosophy, which doesn't mean I'm not in favor of it, but I tried to keep away from philosophy. Schrödinger sort of began to float away a bit, I'd say, at times. And others have written it. I think Haldane did as well. But anyway, but I tried to keep away from philosophy. I tried to keep it simple, and I tried not to be too exciting. You know, so that actually you could pick it up and, you know, it's such a lot of effort writing something. You know, when it goes, you know, when it stops being readable after 5 years because it's all wrong, I just didn't feel comfortable with that, you know. You could read this in 50 years and you'd say, well, you know, it was sort of all right. And you could say, well, that just means it's boring, and that's partly true. But it's at least it's more interesting to read than an undergraduate textbook. I realize that's a very low bar, but I mean... There were more somewhere. Tell me when to shut up.
So when most people talk about finding life on other planets, they're talking about the search for water.
Yeah.
However, you don't mention water or fluidity, so are people looking for the wrong thing, or?
No, water's fine. And I said, solution chemistry in cells, which allows you to rewire. So I didn't use the word water, but I think I used the word wet, which I suppose is wetware, which is close to it. If we're going to look for life in this solar system, the best way to start is with water. We used to think it might be Mars, but it's very dry, and now we're getting excited about the planet, the moons of Jupiter and Saturn. And that is amazing because the geothermal activities keep liquid water despite the distance from the sun. So it will be very, very exciting. These satellites now going there which might catch plumes of water coming out. I mean, just maybe there'll be something there. Now, let's imagine, is it going to be built the same way, or is it going to be different coding, I mean, this would be truly amazing. And it is beyond my imagination, at least, to imagine anything different, although I've said the principles could be delivered by other chemistries, which I think in principle is true. I sort of think, with my feet on the ground, it will be the main thing we should think about is the life variants on what we have, which could have different coding and so on, and water is critical. Something else I want to say about water. Something that has really quite surprised me. The solar system's 4.4 billion years old. So the Earth formed about then. It took about 300-400 million years to cool down for water to exist in aqueous form, liquid form. And yet the first fossils we see are probably 3.7 billion years ago, 3.5. In other words, we went from chemistry to life in about 300-400 million years. And yet to get multicellular life, we needed another 3 billion years. Now, I'd have thought getting any life is the hard bit. Once you've got a cell, you could make us. I have no problem with that. But getting the first cell, I do have a trouble with. Yet it seems to have occurred in a few 100 million years. I'm sure there are. I of course could speculate, but there's no solution to that I know of. So the can you just say it again so other people...
Does that last point you made make you wonder about the panspermia hypothesis?
Well, it makes you wonder about two things, doesn't it? Panspermia is, for those who, is that life arrived fully formed as a single cell from some other system, although the nearest stars are quite a long way away, and I mean, probably would have been at that time. And of course, the other is that there's a divine creator.
But I have something a little in between as a possibility.
Yep.
So even though our solar system may be 4 billion or so.
4.6 billion.
The universe is closer to 14 billion.
Correct.
And we've had a few equivalents of our solar system cycles happening in our own galaxy since then. So if you think about that, if some very, if a lot of life came up all over the place, even rare, but there's a lot of place there, then maybe it was just sent here.
No, Rod, I've got sympathy for it because the 300 or 400 million years just seems ludicrous to me. And when I first read about it, before I started thinking about the time, I thought, well, this is just, balmy because, it came in on a comet or something from somewhere. But I think we've got to imagine these things because it is a mystery. It is a mystery.
I just want to add one caveat here, which is that the textbook presentation for why you have to wait 3 billion years is that you're waiting for plants to terraform the planet, right? You're waiting for the oxygenation of the atmosphere, and that can take a lot longer than the evolution of any individual organism.
I don't quite buy that, though. I know the argument. I think that it is an important feature to think about, but I think to make multicellular life, there would have been ways around that. I mean, I know I'm a bit out on a limb on it. I'm struck by 300 million years to make a cell, another 3 billion cells to make, you know, just a few cells acting together. Doesn't seem... We'll find a way, yeah. Was there something at the back? Somebody at them? No? Do you want to stop?
Oh, I think that's probably a good place to stop.
Yeah, good. Thank you very much. Thank you.